So you guys, this unit has all been about proteins, protein structure, and how they work. Today we're going to be talking about the extracellular matrix, which, as Tom might have intimated to you, is the bread and butter of his lab. So his enthusiasm has extended a little bit to me, but his pet molecules are not mine, and I'll give you a little bit more about my favorite ECM components. And we're going to couch this in tissue engineering and regenerative medicine because ECM is basically all that matters when you're talking about biomaterials. So what are tissue engineering and regenerative medicine? Tissue engineering kind of evolved from what we consider like base biomaterials. So you think about joint replacement. So figuring out that titanium is relatively biologically inert, that was a huge thing. But it's not something that's actually interacting with the host body. It's more of not causing the host body to actively reject it. And those are sort of the baseline of biomaterials. And as you want more and more sort of physiologic outcomes for wound healing and injury, you don't want something that can just be passively tolerated by its recipient organism. You want something that actually integrates and can return into functional constructs, so restoring, maintaining, or improving damaged tissues or whole organs. So the best we've got right now is artificial skin and cartilage. They've got FDA approval, but it's really difficult, as you might imagine, and I'll go through it for any kinds of biomaterials to actually get approved, because it's not just a drug. It is a material, so it's an implant, and then it's also a drug, and then it's also like a disease-causing agent in and of itself. So there's a lot of regulatory hurdles that need to go through before you can even think about getting a product in people. And at any point, if any of you have any questions, I think Tom normally has the policy. You can interrupt or raise your hand. Please do so. So what is regenerative medicine? How is it different than tissue engineering? Regenerative medicine seeks to more exploit the endogenous wound healing and regenerative capacity that you have. So if you get a cut, you don't have to go buy some super expensive synthetic skin. Your skin's pretty good at going over the wound itself, and sometimes you get a scar and sometimes you don't. We'll talk about that. But it's engineering materials and sort of as a discipline trying to discreetly design things that leverage the body or oftentimes trick the body into sort of acting like it's a fetus again. And fetus, so a lot of like umbilical-derived stem cells, the developmental sort of cell niche is a lot more regenerative and a lot more able to heal things properly without scarring than adult tissues. And lots of the fibrosis tissues that are based on ECM are primarily diseases of aging. You generally, as you get older, are less and less capable of successfully healing without functional loss. So these get kind of conflated, and you'll see them bandied about a lot, and nobody really cares too much about splitting hairs, but we figured we'd introduce both terms at the beginning of this lecture. Because again, the pie in the sky is not, you deal with a scar, you get plastic surgery, get rid of your scar. We'd like you to not have a scar in the first place, or we'd like you to be able to heal your fibrotic lung instead of requiring a lung transplant. All of these things are sort of terminal and require replacement. We want to figure out how we can go from replacement to you fixing yourself, basically. So all that said, the biggest thing that really, really matters is the extracellular matrix when you're thinking about biomaterials. So it's a very broad definition. Proteins and polysaccharides, so sugars, polymer sugars, that are locally secreted by cells and assembled into an organized matrix or mesh. So there are tons. This is going to be, like most of these lectures, very superficial. Most of the figures are from, I'm sure Tom's already talked about this, Albert's cell biology textbook. It's kind of the Bible for cell biologists, even into the graduate level, and I love it dearly. So if anything really tickles your fancy, you can come talk to me or send emails, or just pick up a nice 500-page textbook. Main classifications that we're going to talk about today are basal lamina, otherwise known as basement membrane, and connective tissue. So real quickly, the basal lamina, you'll find it underneath or separating different areas of tissue. The most classic is epithelium, so your skin, or say the lining of your gut or the lining of your lung. And underneath that epidermis for your skin is the basal lamina. And what it does is it also provides polarity to these epithelial cells. So when I say polarity, I mean at the top you have what's called the apical surface of the cell, and at the bottom, touching the basal lamina or the basement membrane, you have, as you might guess, the basal layer of the epithelial cell. So in addition to structure, basement membrane also provides cues to the cells to know which way is up, basically. And underneath that, you've got the connective tissue. So this is a mess of bioactive saccharides and a lot of fibrillar proteins that we'll all talk about. And you can see here, this is an SEM of what the basement membrane looks like. So on the top, you've got epithelial cells, and then you have a really thin sheet, and that's all the basement membrane is. But it has a bunch of different varieties, and it has so many different biologic functions. And then underneath the basement membrane, you can see collagen, which is one of those connective tissues we're going to talk about. So what physiologic roles does extracellular matrix play? And the answer is all of them. So structurally, you're not just a pile of cells, because if you were, you'd kind of just be a puddle in your chairs. All of the organization of your body, every tissue feature, your eyes, your lungs, all of it is dictated by the extracellular matrix structure and organization. The amount of cues that cells receive from the basement membrane or the extracellular matrix can't really be overstated. So structurally, in terms of like physical things, engineering things, tensile strength, compressive strength, loading, torsion forces, all that, that's what ECM does. So cushioning and cartilage, the ability for you to actually jump up and down and not have your bones just smashing into each other and breaking apart every time you run. Actually using the basement membrane as a filter. So the basement membrane itself is in certain organs has a pore size of such that it by itself, not structurally, but just because the pore size is a certain size, won't let certain molecules through, but will let other molecules through. We'll talk about cryptocytes a little bit, and then at the cell level, so like I talked about before, cell polarity, cell adhesion, so cells adhering to both the extracellular matrix and to each other, morphogenesis and differentiation. A good example of that is, so laminins, this other table shows phenotypes of mouse lines that were generated to be deficient in specific ECM components. You'll see that with the laminin and fibronectin and collagen, they're all just lethal. Once you have two cells, once you've done your first division, the zygote, you have laminin. And if you don't have laminin, it won't make it past 4 cells. So these proteins are absolutely required for cell development. So cell migration, cells use ECM to sort of know where to go. They actually can transform the ECM that they live on into sort of highways, and then a leader cell will bend the ECM into like a straight line, and then all the cells following it will have a much easier time. Proliferation, so depending on where the cell is, sometimes it's useful in wound healing for the cell to proliferate, and then it's also sometimes very important for it to stop proliferating. Can anyone think of a disease where cells keep on proliferating when they're not supposed to? There you go. So not being able to sense extracellular matrix cues is actually a hallmark of a huge number of cancers. So, and then prevention of apoptosis. So this is another cancer thing. So either preventing the cell from killing itself or telling the cell, okay, it's time to stop. Like you can program cell death. So basically every structural and structural component of your body at like the macro scale, and then every cell behavior to some degree can be influenced by the ECM. Like I talked about before, we're going to talk about two big things, the fibrous proteins and the saccharides. So fibrous proteins are also known as glycoproteins, and then glycosaminoglycans, or GAGs. And these are bioactive saccharides that are commonly associated with a core protein, which are called proteoglycans. And I know proteoglycan and glycoprotein are going to be a little confusing, but hopefully I'll repeat it enough that we'll be comfortable with it at the end of the lecture. So some of the fibrous proteins are here in green. Laminin, collagen type 4, type 1, nitogen, fibronectin, and then here are your saccharides. So this is, the saccharides are in pink. So perlecan, hyaluron, decarin, and aggrecan. So you can see some of these have their proteoglycans, these little green things that they're attached to, but the vast majority of the molecular weight is the sugar, and the protein kind of exists to let it interact with other molecules or to organize the sugars. So getting into GAGs, again, it's if you've taken any kind of biology recently, like AP Bio or something, disaccharide repeats. These are just sugars, kind of like a modified starch. So you've got an amino sugar, so it's just a sugar with an amino group on it, and then an acidified sugar. And they're classified based on the type of sugars that they are and the type of linkage, so which carbons are bound to which, and then the number of location of the sulfate groups on there. So you've got hyaluron or hyaluronic acid, chondroitin sulfate and dermatan sulfate, heparin sulfate, and keratin sulfate. Keratin, hazard guess, anybody where it might be? It's in your skin. So when people talk about keratin, keratin sulfate is also associated with those sort of very protective layers. So it's something that gives skin its elasticity. So you stretch it, it doesn't stay stretched. So like with all proteins, like we've talked about, the structure kind of dictates the function. So the conformation of these GAGs, a lot of them are huge. So I think you guys talked about proteins being in the kilodalton range, like you get proteins 100, 200 kilodaltons. GAGs can get up to megadaltons. So these guys are huge polymers. They have a huge volume to mass ratio, and a lot of that is because of their charge. So almost all of the GAGs are very hydrophilic to a point that as a bulk, they kind of absorb water from their environment, and that's something that's called hygroscopic. So these guys want to swell, and all they want to do is just fill all of that negative charge with water. And so you can imagine what this does is if they're embedded in a matrix that doesn't let them swell, they're going to constantly be exerting a passive tension on the tissues that they're in because they're constrained by the rest of the body. You obviously don't stand in water and swell up like a sponge. So the sort of passive tension of swelling pressure is called turgor pressure. And because of this, the GAGs are super useful for high impact things. So they provide these compressive forces. So some GAGs you'll find in joints, and I think we'll have a diagram in a second. Oh, no, we won't. So you can imagine your knee joint. It's one of these GAGs in the articular cartilage there, and as you put pressure on it, the water is forced out, and the force of equilibria of the water wanting to go in gets higher and higher and higher the more compression you have, and eventually that force is so strong that the water has to come back in. So it's a force and load buffering system, just from the fact that it wants as much water as it can possibly get. So here's a couple again. Like I said, they're super useful as a lubricant or a shock absorber for most of the joints in your body and all the tissues that need to move past each other without abrading and getting injured. You don't need to know specifically the unique features of these. Now we're going to get into the proteoglycans. So the proteoglycans are kind of paradoxically enough more about the sugars. So these are the small proteins that associate with a lot of the GAGs. These aren't the big fibrous proteins. Those are called glycoproteins. So I guess focus just on the suffix here, on these two words, to not get them confused. So with these, you've got a protein backbone, which we can see here, and then you've got your sugars attached. So to produce these, you've got a core protein. Like most proteins, like all proteins, it's made in the ribosome and then trafficked to the ER, and then the GAGs don't actually get added until it's trafficked into the Golgi, and then it's exocytosed from the cell into the local environment, where it'll then swell as much as it can with water and do its thing. So proteoglycans, they can be smaller. So decarin is a tiny sort of, you can consider it like a bridging proteoglycan, whereas aggrecan is a monster and can get up to, that's one of those megabase glycoaminoglycans. And you can see here a really high magnification. It wants to get as straight as it possibly can, because if all the water is in there, there's not going to be room for it to wiggle around. But in your body, it's all tangled up. And so that's, again, what provides the turgor pressure. So what do they do? We've talked about it a lot, but let's reiterate. They contribute to ECM assembly and organization. So decorin is one of the things that sort of joins other larger ECM components together. They regulate traffic of molecules in cells. So like I talked about previously, ECM, even based on pore size, can regulate the availability of molecules and diffusion rates. The swelledness of the ECM regulates how quickly small molecules, water-soluble molecules, are going to be able to diffuse through it. And also the charge, so the available charge on the network will obviously impact the ability of polar or non-polar molecules to get through. Because of this charge and because of just the sheer amount of protein, they also have a propensity to sort of sequester chemotactic signals. So all of these signaling molecules, they can actually be secreted and then taken up by the proteoglycan network, and they kind of just hold on to it. And if they ever get degraded, it's locally available to the cell that degrades it. It doesn't have to migrate in from circulation. They're already there in the tissue. Oh, no, we do have an example. So aggregate and cartilage tissue. So this is a diagram of what I described before. So normally, you've got this nodule of cartilage, and it's swelled passively. And then when you put pressure on it, water is forced out from the force. But as the compression and as the more water is forced out, the sort of osmolar gradient gets higher and higher and higher. And at some point, you can't compress it anymore. It's like getting two magnets really close together, and the water will come back in, and the tissue will rebound elastically to where it was before. So that's kind of it for the proteoglycans. I wanted to get those out of the way. They are super important, don't get me wrong, but most of what our lab looks at is some of the more fun, longer proteins and less of the bioactive saccharides. So we're going to jump right into it with the basement membrane, which is one of those two big membrane components, or ECM components, excuse me. So how does basement membrane look? It underlies the epithelium, it surrounds cells, or it can be found between sheets of cells. Yeah?
Hi, sorry.
You're good.
I'm like slightly lost. Can you go back to the last slide?
Yeah.
And maybe one slide before. So can you just reiterate how does it regulate the traffic of molecules in the cell, and then like the whole osmolar gradient?
Yeah. So for trafficking, you can imagine it's just a, what do we call it, allosteric hindrance. I think you guys talked about that previously. So basically, literally, there's no room. So whereas the fastest path for a molecule would be diffusing in a straight line, you've got all of these saccharides and proteins swelled with water in the way. So it's going to be bouncing around. So the more dense these molecules are in the local environment, the longer the diffusion time from point A to point B will be. Does that make sense?
That makes perfect sense.
Yeah.
Actually. And the chemotactic signal, I'm not really sure that word means.
Right. So chemotactic signals is in this it would be something that's going to cause a cell to want to follow it. But there's plenty of water-soluble molecules, so like growth factors or danger factors like interferon gamma or fibroblast growth factor is a growth factor. So these can actually be held locally within this sort of swelled hydrogel that's made up of these saccharides. And what that means is they don't actually diffuse anywhere. So you can imagine if the signaling molecule is superpolar and the proteoglycan network has a complementary charge, it's just going to want to stay there. And that signaling molecule will stay there until a cell comes in and starts chomping on things. And then it'll see the signaling molecule. So it's a way for local sort of storage of all of these signaling molecules, as opposed to they might have only a half-life of a couple seconds if they're floating around, because another cell will see them almost immediately.
Gotcha.
Does that make sense?
When you like, get an injury or something, the histamines flowing to that area,
Mm-hmm.
you put histamines there to elicit like a larger immune response. Is that kind of an example of this?
I would imagine it happens. There's not very many molecules that are immune to sort of mass transfer in like the laws of physics. So yeah, I couldn't tell you specifically does histamine get accumulated locally, but
That's really possible.
yeah.
And, sorry, one more question. The whole osmolar gradient thing you're talking about in the next slide, why does it kind of rebound, or why is it at lower rate?
So what's happening is you can see this backbone, it's getting all crumpled, and it's getting crumpled because there's less water there taking up the volume and there's force pushing it together. And what it means is now you have these polar sugars getting pushed up against each other, and they don't like that. They want a water to buffer that charge. So as more and more of these, it's like I said, pushing two like-sided magnets together, they're really going to resist that, and they want to bounce back apart. And so the net charge differential between inside and outside is going to get really high, and it's going to want water to shoot back in. Does that make sense?
Yeah, that makes sense why water won't come back in, but what would cause compression in the first place?
So in this case, this is cartilage. So like you can think about your knee. You're literally just stepping, and you're pushing water out of that hydrogel.
Gotcha.
And the reason it rebounds and you're not just like grinding your bones into each other is because of that rebound. So it's not like this expendable sheet that every time you step gets destroyed. It's a nice little it's like a rubber ball. And as you get older, the actual composition of this proteoglycan mesh can change, and you get less sugars, I think, and that actually is cause of some connective tissue disorders, and that's why your joints start to hurt, because you don't have that buffering capacity anymore.
Thank you. That helped tremendously.
Yeah. And anybody else interrupt me. I know I can go fast sometimes, but we do have a decent amount of content to get through today, because I think last year this was 2 lectures. I cut a lot out, so don't get too worried. Any more questions about GAGs and proteoglycans? Okay. So basement membrane underlies the epithelium, surrounds cells, or it's between 2 cell sheets, and that's kind of dependent on the type of tissue we're talking about. And like I said before, the lumen external surface, or apical side of a cell, and it's important you know apical versus basal, is directed by the basal lamina or the basement membrane. I'm going to try to keep on calling it the basement membrane, because only really veterans of the field, you can put it that way, still call it the basal lamina. It's kind of a older term. And then underneath that you have connective tissue. So in muscle, you've got muscle fibers, and then around each sort of small bundle of muscle fibers, they actually have their own sheaths of basement membrane that connect everything together. In the kidney, so I talked about earlier, one of the functions of the basement membrane is actually to act as a filter, like your coffee filter. It has a very specific pore size that your kidney takes advantage of for filtering and generating urine from blood. And if this basement membrane gets disrupted due to, say, a mutation in one of the proteins in it, you can actually have severe kidney problems. So here's a bit of, we're gonna talk about 4 main components, and the 2 big ones I want you guys to focus on are laminin and collagen 4, both of whom are near and dear to my heart. And then we'll also talk about nitrogen and perlecan, but they're not as exciting. So you've got a cartoon here. This sort of gray background is a cell surface, and then you can kind of see the network architecture of the basement membrane. So it does make a very thin sheet. But I just want to emphasize on scale here. You guys talked about the scale of cells. Does anyone know how big a cell can be? 10? Yeah. So just pay attention here. This is orders of magnitude difference. So these are 40 to 120 nanometers. So these are on length scales of hundredth to smaller of the thickness of a cell. So they're very thin compared to the cells on them. So they're these very thin sheets. And you've got the big structural components of this network are going to be laminin and type IV collagen. And then we'll see that perlecan and nitrogen are kind of what bind one mesh network to another mesh network. So they're kind of the cross-linking agents that actually let the meshes sort of work together and not just be 2 separate meshes moving around. So if anyone's squeamish, I would advise, I'll tell you when you can look again. So this is going to be some disease relevance, because that always gets things exciting. If any of you have heard of epidermolysis bullosa, you know it's nasty and it's not fun to look at. So as the name implies, epidermolysis. Does anyone want to take a guess what that means? Sure. Exactly. And bullosa just means essentially boils or like blisters. It's not a fun disease. And junctional refers to a very specific cause of the disease, which is the inability of the epidermis to adhere to the dermis because of deficiencies in the basement membrane. And it's caused by two things: the laminin itself being mutated, or integrins, which we'll talk about later, are what cells use to bind to laminin and other ECM components. So these are some fluorescent images of the epidermis and the dermis in normal skin, and you can see between them you have this wonderful thin basement membrane layer of laminin that they labeled with a fluorescent probe. And then this is a case study, and the cool things you can do with genetic engineering, there's nothing here. So this patient had a birth defect where his laminin wasn't able to form a proper basement membrane. And at admission, and again, warning, look away if you're squeamish. So at admission, this kid was 7 years old, weighed 15 kilograms, which is low 30s, and had skin on somewhere between 5 and 12% of his body. Everything else, no skin at all. They had tried everything. So they tried the FDA approved, this was in Italy, so no, they're probably still FDA approved. They tried the artificial skin. They tried skin grafts from the father, didn't take. They had kind of given up, but his parents kept on asking if there like anything they could do. So they took some skin from this kid and they virally transfected. So what they did was they engineered a virus a retrovirus. So those are the type of viruses that actually permanently insert genes into the cells that they infect. They engineered a retrovirus to have the non-mutated laminin. They infected the skin cells that they had took. So these are skin cells in a dish, his skin cells in a dish. They infected it with the virus. They inserted the proper laminin gene where it needed to go. They grew them into massive sheets. And then they grafted his own skin that had been virally transfected back onto him. So this is him after a couple months. And he's got functional skin coverage over about 95% after, I think this was 3 months. And this was in 2014 or 2015. And now he's like 11 or 12, and he's doing great. And this is what the skin looked like after. So complete restoration in these skin grafts of the laminin. So this is the kind of, when we talk about why these molecules are important for basement membrane, super, super, super important that it works properly. So let's get into it. And yeah.
Not to this one, but the one before that, this one. When it talks about the rigid cross-linked sheet, is that just talking about the interactions between the different proteins, or what exactly is that?
Yeah, we'll get into it more a little bit, but I can tell you, so collagen 4 makes like a mesh, and laminin makes another mesh, and by themselves, they actually aren't bound to each other, but they're both super vital components of the basement membrane, and nitrogen and perlecan are the things that cross-link those two sheets together. So laminin is a bit of a complicated molecule. It's a heterotrimer, which means it's got 3 components, and each component is different. So you have an alpha, a beta, and a gamma chain. There's a bunch of different isoforms. We're not going to get into all of them, and you're not going to be expected to know all of them. Laminin 1 generally refers to laminin 111, and so this 1 would be alpha chain 1, beta chain 1, gamma chain 1. So you can imagine if even there's 2 versions of all of each alpha, beta, gamma, the permutations go up pretty quick. So like I said, it organizes the basement membrane. The key things you need to know are it has 2 short arms with the beta chain and the gamma chain that go up to the side, and these facilitate the self-assembly of laminin into that mesh-met structure. And then the long arms on both the N and C terminal domain facilitate actual interactions with other cells. So we'll talk about integrins in a little bit, but they have epitopes that cells can actually recognize and bind to. And this is what the actual molecular structure looks like. So it was through X-ray crystallography, and I think this is a TEM, they were able to verify the structure. So the next one we're going to talk about is collagen type 4. So laminin's about 50% of basement membrane by mass. Yeah, question?
What did you say the function of the long arms were?
So long arms facilitate integrin or cell binding. So integrin is cell binding. Integrin is a protein that we're going to talk about in a little bit that's on the surface of your cells that allows your cells to bind to extracellular matrix components. So collagen type 4 is the other half by mass, which is kind of cheating because nitrogen and perlecan are tiny mass-wise, but by weight, collagen 4 and laminin are basically 50-50 of all the basement membrane proteins. And as with all collagens, which we'll talk about more later, it's got 3 alpha chains all wrapped together to form a triple helix. And all collagens are characterized by Gly-XY repeats. That's what gives them their twist. And what these do, and you can see, it looks like a rope, and that's exactly how it functions. So they have extremely high tensile strength. But as you can imagine, if you were to try to push a rope together, it's just going to coil up and provide no resistance. And that's exactly what collagen 4 is like. So you'll find that most of the ECM long proteins provide tensile strength and resistance to pulling, and all of the hydrogels, saccharides that we talked about, provide the turgor pressure, so the outwards. So they're always at an equilibrium, and there's constant tension in all of your ECMs between the swelling forces and the tension forces. Let's see, excuse me. I know, it's riveting. So nitrogen is one of the small guys. It's a glycoprotein, 2 isoforms. And like I said, it'll bind laminin and collagen 4 together. So it's kind of one of those cross-linking agents with a bunch of other molecules too. Fibronectin, we'll talk about in a little bit, is the kind of ECM component that basically just lets cells know what's going on. It's less structural and more of a functional readout for cells. Fibrinogen is the key protein in clots. And like I said, it bridges, the thing you need to know is it bridges laminin with collagen 4. So perlecan is another one, also bridges laminin and collagen 4. It's also found in connective tissue. And again, it binds nitrogen, collagen 4, does not bind laminin, will also have epitopes for integrin binding, and heparin, which is another one of those GAGs. Yeah?
So when it says like heparin, is that related to like the blood thinner heparin?
Yeah, it's actually it's the same thing. So heparin is everywhere, and local heparin is actually an anticoagulant. And it's good because you think like blood it really wants to clot. So heparin sulfate is a wonderful molecule to make sure that you're not throwing thrombi as you walk around in your day-to-day life. So how does it assemble? The short answer is most of these self-assemble. The long answer is it's probably way more complicated than that, but we don't really know. What we do know is that you've got, and I think you guys have talked about these before, proproteins. So these are proteins that aren't in their functionally active state yet. Can anyone hazard a guess why you wouldn't want a self-polymerizing fibrous protein to go in your cells? Yeah.
You don't want to polymerize and leave your cells?
Correct. Yeah, that wouldn't be good for your cells. So they are assembled in the Golgi apparatus, and then they're secreted, and once they're secreted, either through integrins or spontaneously, they self-assemble into these mesh networks. So both laminin into its honeycomb structure and collagen 4 will self-assemble once they've been secreted. And then they require those 2 smaller linker proteins to sort of make a full mesh. Yeah?
What are district glycans?
So district glycans are a bunch of, so they're these guys. They're a way that cells have to bind to ECM components. We're not going to be talking about them today. We'll mostly be talking about integrins, so don't worry too much. But it's just another, cell has, pretty for everything you learn in this class, there's about 4 other things that do the same thing that we're not covering. So another functional readout, or functional use for, so in this case, this is a laminin. Integrins are what are used to sort of sense ECM components. This base of the laminin has these 5 modules on them. You don't need to worry about what they are. But in this conformation, there's a specific integrin that can bind to it. And this integrin tells the cell to migrate. It tells the cell to scoot and secrete matrix and move. And this site is sort of made available to the cell in injury. So the cell will start moving towards the site of injury. And then once it gets there, plasmin, which is actually a serine protease involved in clotting, will increase in local concentration as you get closer and closer to the wound. And once you'll reach an equilibrium where plasmin actually starts cleaving most of these bits of the laminin. And what this does is it changes the availability of integrins on this laminin and allows alpha-3 beta-1 as opposed to alpha-6 beta-1 to bind. And what alpha-3 beta-1 says to a cell is stop migrating and start attaching to everything. So start laying down roots, basically. Make cell-cell contacts, make cell ECM contacts. You don't need to move anymore. And what this is called is cryptic site. So this original conformation doesn't let alpha-3 bind, and then once it's cleaved, once the molecule is cleaved, this cryptic site is now exposed, and it changes the behavior of the molecule as it interacts with cells and other ECM components. So cryptic sites are a huge thing in cell biology. Let's see. So that's it for basement membrane. Any questions on basement membrane before we move on to connective tissue? Good. So connective tissue is kind of the rest of the ECM. It's not a thin sheet like basement membrane is. So for location, beneath and surround. So pretty much everywhere else that's not basement membrane or cells is going to be connective tissue ECM. And this is primarily what the Barker lab focuses on. So components, protein fibers embedded in a amorphous mixture of large proteoglycans. So it's more fibrous protein, but this time instead of a thin sheet, it's a much more three-dimensional structure together with all of those bioactive sugars that we talked about that are swelling. So it's everywhere. There's more of it by volume than your cells, and the composition and organization of this connective tissue ECM determines the physical properties of the tissue. So we can't talk about connective tissue without talking about fibroblasts. So fibroblasts are the predominant cell type in connective tissues, and we call them fibroblasts. They are the ones that secrete the fibers. Or in the case of aberrant wound healing, make fibrosis. So if you hear about fibrosis, it is an excess of fibroblasts that don't calm down. So they secrete all these ECM components, the fibrous proteins and the GAGs and their proteoglycans. And again, to give you this cartoon, so we're done with the basement membrane here. All in here, you've got collagen fibers, elastin fibers, a whole mesh of proteins and saccharides that provide the structure and organization to the rest of the tissue. The big one, we already talked about collagen 4, now we're going to talk about them more generically, is collagen. So it's the most abundant protein in your body. If you were to be broken down to your constituent proteins, it would be about a quarter of your weight. So the proprotein or procollagen, before it's secreted and sort of organized into fibers, is 3 alpha chains. It's not an alpha helix, just to emphasize, it's an alpha chain. And each of these monomers, each of these alpha chain monomers combine into sets of 3 to form a triple helix. And like we talked about with collagen 4, all collagens are characterized by repeats of glyXY. X is usually a proline, and Y is usually a hydroxyproline. Does anybody know, huge kudos if you do, why hydroxyproline is important and what happens without hydroxyproline? Okay, I'll give you a series of hints. First hint, you need ascorbic acid in order to add hydroxyproline to collagen. Ascorbic acid is vitamin C. Can anyone think of a disease with vitamin C? Yeah, so vitamin C is required to have this post-translational modification onto proline into hydroxyproline. And without it, your collagens are not stable at body temperature, which is why people with scurvy bleed from basically all of their mucous membranes. And if it goes long enough, they essentially fall apart, as you can imagine you would if none of the 25% of your protein could actually hold itself together. So there are a dizzying number of different collagen chains. And so there's 42 different alpha chains, but we've only seen 28 combinations in vivo of them. To get a little bit more into the structure, said it's a triple helix. So glycine is very small. Yeah.
So can you go back to the last one?
Yeah.
So what exactly makes it like an alpha chain? Like what is like a defining chain? Is it just like YXY?
Yeah. So the chain is It doesn't really have any basis in terms of like crystallography or structure, but the GlyXY, and we're going to talk about how it twists in the next slide. So glycine, as you guys talked about in the amino acid, it's smallest. It forms the interior. So you can see here that the glycine on the inside, because there are no residues, there's nothing crazy going on with the glycine, so it's going to stay on the inside. There's no side chains. X is usually proline. And we know that prolines have a really sharp bend to them. I think you guys talked about that. And that's what provides these really sharp turns in each individual helix. And then hydroxyproline is this modification is required to provide stability, further stability to the collagen molecule. So that's how you get these tight curves. It's basically the two prolines give it a really sharp turn, and then it has the glycine to sort of hold everything together. So like I said, there's a huge diversity of collagen. You've got fiber-forming ones, and you don't have to memorize all these, so please don't worry. But just know that there are fiber-forming collagens. There are fibril-associated, so they're fiber-like, or they associate with other collagen fibers. Collagen 4 is found in the basement membrane, like we talked about. And then there's some newer ones, and I feel like they make a new one every week. Transmembrane and proteolytic core proteins. So they were first identified as these long fibers, and then someone figured out that their structure was GlyXY. How do you think, what would you look for if you found something in like the transmembrane? How do you think people would find, like, how would you know that you're looking at a collagen if it's stuck in the membrane? It's not just like this nice rope. Yeah.
I have a question about like 2 slides again.
Yeah, you're good.
So when you said that it's 3 alpha chains, but it's not an alpha helix, but it forms a triple helix, what's the difference?
So an alpha helix is like a very conserved definition of a sort of ribbon going like this. This is not that. This is turning too tightly to be that. These are called alpha chains. I don't know why. I can't tell you why. They are just called alpha chains. And because there are 3 of them, it's in a triple helix. I don't know the structural definition of an alpha chain. I just want to emphasize to you to not conflate the 2. I'm sorry, that's an unsatisfying answer, but that's all I've got. Okay. We'll just move on for the sake of time. So how it actually turns from individual triple helices into collagen fibers that provide, basically, they look like a rope. If you've ever seen a diagram of a rope or even a muscle, you'll see this sort of successive, almost fractal stacking in a lot of biologic fibers. So you've got an individual alpha chain, then you've got your procollagen, which is an individual triple helix, and then fibrils, which are tiny fibers, which are several, so that's what this guy is right here, which are several individual procollagen molecules together, and those all together make a fiber, which can get up to several microns in diameter, and they can provide a massive amount of structural strength. So yeah, getting into what they actually look like, your tendon here, the form always follows the function. So tendons, because they basically provide uniaxial tension force, they're only going to be aligned in one direction. They only really need to provide force in one direction. However, things like bone, or in this case tadpole skin, which actually has a structure similar, you've got alternating layers of fibers going this way, and then these little circles are actually fibers coming out sort of towards your eyes. So you've got sort of orthogonally or perpendicularly aligned bands of fibers, and that provides the ability to resist pulling in all sorts of different directions. So how does it actually get stabilized, and what makes them not fall apart? Like, how do you go from an individual strand to a rope? Like, what makes that a cohesive whole? Anyone think of what lysyl oxidase might do? What's the function of this enzyme? Good, you all whispered it. It oxidizes. So what it does, as you guys might have already talked about, you get aldehydes. And aldehydes are super reactive. They don't want to just be left alone dangling on the edge of a molecule. So lysyl oxidase will take lysines on the external bits of collagen fibers and add the aldyl groups to them. And what that wants to do is then react with like. So it'll either find another aldehyde group, or it can even sort of spontaneously bind with a unmodified lysine on another collagen. And whenever we have covalent bonds being formed between different molecules or different polymers, we call them crosslinks. I've said that a lot this lecture. So what provides the ability of these tissues to sort of return? Elastin is the sort of big elastic molecule, as you might guess from the name. So its common repeat is VPGXG. So there's no glycosylation or hydroxylysine here. It has prolines on it. Tropoelastin is the soluble precursor. So again, you don't want fibers being formed before it gets outside the cell. And it cross-links in a similar way to collagen does with those oxidases. So it's got, what gives it its properties is it's got the hydrophobic regions, which we talked about previously with the sugars, provide the ability to want to return to your normal state. And then it's got alanine and lysine-rich alpha helical segments. And that lets, the lysines especially are very amenable to all sorts of modifications and binding. So aside from arteries, can anyone think of other tissues that need elastin? What do you stretch all the time? Skin, I heard muscle. My pet organ is the lungs. Your lungs need to be able to expand and contract millions of times in your lifetime. And the ability to do this is because they're full of elastin. And without elastin, you'd breathe once and that'd be it. That got a response, that's good. So talking about fibers, how elastin's organized, you've got an elastin core, and then it's glycoproteins, fibrillin, which binds to elastin. Yeah, question?
Could you explain one more time how like the hydrophobic property or the hydrophobicity gives it elastin?
Right, so if you think of it as a fiber and it's getting pulled, again, those hydrophobic residues, because it's going to be structured as a fiber, really don't like being next to other hydrophobic residues. It's putting two negative charges together. So just based on like reduction in free energy, entropy kind of thing, it's going to want to avoid that interaction as much as possible. So that's it. It's just charge reduction. You want a net zero charge in the system.
I have another question.
Yeah.
So like before when you talked about like the compressive forces and like wanting to expand, that was due to hydrophilic properties, not hydrophobic properties?
Yeah. So for the bioactive saccharides, the reason that they swell is because they want water in them. In this case, the reason these guys, if you stretch them out, want to come back together is because in stretching them out, you're actually compressing the fibers together, and those hydrophobic residues are getting closer and closer and closer. So it's not specifically hydrophilic versus hydrophobic, it's just hydrophobic and hydrophobic together is a lot of the force. So it's like and like. It doesn't have to be just hydrophilic or just hydrophobic. And then, yeah, we got microfibers. So here's what it looks like. Normally, these elastic fibers are all bundled up together because they want to be all curled up. And then you stretch them out, and all these hydrophobic residues get exposed that they don't want. And so it's going to want to return to its bundled up state. Does that make sense? And this is a scanning electron of what elastin looks like when it's stretched, actually. Let's see, how are we doing? We're good. We might actually get done a little ahead of time, so we'll have ton of time for questions. I shouldn't promise that before we're done. I might spend so long on fibronectin. So fibronectin is one of the smaller fibrous proteins, and like I said previously, it's less structural and more about informing cells and giving them cues and ligands and signals to act on. So we talked about heparin before, but we've got cell binding domains, collagen binding domains, self-association domains, so fibronectins can form their own network. This cell binding domain, which is expanded here, if you take any or look at any biomaterial, this RGD sequence is key for almost everyone who's trying to make a biopolymer that cells can bind to. RGD will bind to a huge number of integrins, and it will bind strongly to them. So RGD is kind of the coup for everyone trying to make a biomaterial that actually can interact with cells. Synergy sequence is something that basically increases the affinity or the avidity of the bulk molecule or structure to cells. But RGD is the key player. So like I said, fibronectin mediates a wide variety of cellular processes. So adhesion, migration, all the cell behaviors we talked about at the beginning, fibronectin can modulate those. It's a ligand for tons of integrin receptors. So therefore, it modulates all these behaviors, and then it also it's integrated into the rest of the larger fibrillar ECM network. So fibronectin is never by itself. It's always found as part of the larger whole. But depending on the mechanical state of that network, different parts of fibronectin could be exposed to the cell. So you get differential signaling based on the context, the mechanical and chemical, like pH, temperature, and stress and strain that the molecule is under. You don't need to memorize this, but this is just to give an idea. This is sort of a chart of all of the different things that fibronectin interacts with. So on here you've got ligands. So heparin we talked about. Fibrin is the active form of fibrinogen. So this is the polymer that makes up the clots. Collagen and gelatin it can bind with. It has all of these integrin binding domains here, heparin, and fibrin again. And another thing to note is there are these modules. You don't need to know too much about them. What is useful to know, and we'll talk about again in a second, are the ellipsoid ones, these type 3 repeats. There are a ton of them in the middle, and these, because they don't have disulfide bonds, can actually, in response to mechanical force, be unfolded. The molecule doesn't snap, but these two sides, if they're both attached to things that are moving apart, will actually cause this center region to unfold. And that has huge implications for what cells are able to sense on the molecule. So yeah, that's the force-sensitive region here is all these type 3 repeats. And then within the force-sensitive region, you've got your integrin binding domain. So you can imagine this integrin binding domain is responsive to the forces being put on this molecule. And that's super important for biomaterials. So here's another way of looking at the fibronectin. You've got two arms like we've seen in the other two comics, and then the disulfide bridge in the middle. 440 kilodaltons, and you've got type 1, type 2s, a couple of type 2s, and type 3 repeats in the middle. And then to give an example, so fibronectin display this stretching and unfolding ability. So in their native state, pre-stretched, you've got 35 plus or minus 2 angstroms between this RGD sequence that integrins love to bind to and the synergy site. And then in its full, or not even full, after the first beta strand is pulled apart, that gap has gone up to 85 angstroms. So, and you can see the RGD loop is in a completely different pulled out conformation than it was previously. So what does this matter? How does the cell know that things are being pulled apart or not? Integrins are the big molecule that we like to talk about when we consider how cells interact with their extracellular matrix. So integrins are what connect the cells in the matrix. They're a large family of transmembrane adhesion molecules. It's a heterodimer, so you've got an alpha subunit and a beta subunit. There's, like with collagens, there's a bunch of different alphas and betas. They're combinations, so you'd say like alpha 5 beta 1. There are 24 of those such combinations. There's 24 flavors of integrin. You've got short intracellular tails, and these connect to, this is a hugely simplified diagram. Talin is the big takeaway from this lecture. Talin allows the intracellular part of the integrin to bind to the cytoskeletal network of the cell. But it does so much more than that. This adhesion complex, as it's called, is, as of my last paper reading about 300 different proteins coming on and off. So this complex is super complex, and it does a lot of things in terms of transmitting signals that the integrins generate to the rest of the cell. So it functions as mechanical support, so it anchors the cell. So if you grow up cells in lab on tissue culture plastic, what they're actually using to hold on to the protein that's covering the plastic are integrins. And then signaling, and we're going to talk about the signaling. So how do they work? There's an active and inactive conformation. You guys have discussed allosteric regulation before. So while they're inactive, they're actually not in a shape that gives them a high affinity to bind their ligand. And then once they do bind or detach, they change their shape, and that changes the shape of the entire molecule, so outside in or inside out. It's not unique, but it's very cool in that the integrins can be activated from the inside out. So most of the time when you think of signaling, you think of something binds the external receptor, and then that signal goes into the cell. But something can drive the cell to yank on the integrin from the inside, which causes the outside of it to unfold into its active conformation. So we call that outside or inside out signaling, excuse me. So we'll talk about that a little more and give a brief example. So this is what an inactive integrin looks like. So this is without RGD, and this is with RGD. So you can see it's folded over here, and then it's extended here. And this is recapitulated in this cartoon. So for outside in activation, what you're kind of used to thinking about, the integrin will find its extracellular matrix ligand, bind to it, and that causes this unfolding. And what this unfolding does is it's this really remarkable sort of parade of events, but these alpha and beta tails that are in the cell end up moving apart from each other. And that sort of sparks this whole signaling cascade, because the talins are finally able to bind both sides and sort of propagate downstream signaling. So for the inside out, this time talin competes for the alpha and beta binding sites. And if that event happens, this linkage is undone. So this is the sort of closed conformation linkage. That linkage gets undone, and they're forced apart. And forcing them apart on this side forces the extracellular bits to unfold into their open conformation. So yeah, it drives the extracellular portion into this extended active conformation. And in a feed-forward sort of positive feedback loop, active integrins tend to recruit more of these integrin and adhesion proteins to this site of activation. And so you get more and more signals being propagated through the rest of the cell. So this is called integrin clustering, and it's a phenomenon you see a lot when you do cell culture with fibroblasts. So the matrix is not sort of static. It's constantly being remodeled. It's constantly being broken down and redeposited. You notice this most thoroughly when you have a wound, especially a skin wound, and it heals. So it's being chewed up and replaced. And depending on the balance of proteolysis and deposition, you can get functional tissue versus too much generation of ECM, which gets you those raised, nasty keloid scars. So what you want is a healthy mix of proteolysis and secretion. How do we break these proteins down? So matrix metalloproteinases, I think you guys talked about certain molecules and enzymes that actually need metal ions for allosteric reasons in order to fold properly. So they need those charges. So matrix metalloproteinases, of which there are many, you can see here, again, you don't have to memorize all of them. They're the ones that chew up the vast majority. And then you also have serine proteinases. So we talked about plasmin earlier with the laminin being cleaved, that cryptic site being revealed. So plasmin is the enzyme that revealed that cryptic site on the laminin. And again, to reiterate, the exposure of cryptic sites and allowing cells, especially integrins, to see new parts of the extracellular matrix is a huge cue for those cells. So what they do, they chop up the ECM, they liberate the angiogenic factors. So we talked about sequestering growth factors, angiogenic factors, all sorts of factors are sequestered both in the mess of protein and saccharides. So you can imagine as you chop them up, these signaling factors are now loose and they can actually be sensed by the cell and taken up. Regulation of cell adhesion and migration. So I talked about before, cells will remodel when they're migrating their local matrix environment. So you need MMPs to sort of chew up the crosslinks of this sort of random mesh. And as the cell moves across it, it's actually through force reorganizing the fibers in a direction. So if you have a migrating front of, say, epithelial cells, the first cell, which is called a leader cell, will sort of make a highway on the matrix, and that the rest of the cells, they don't even need to know, they just will follow the sort of straight line path that's made for them by the leader cell. And then lastly, the processing of growth factors and cytokines. So a lot of growth factors actually need to be cleaved by a serine protease in order to become active. So it's another step of regulation of these sort of sequestered or cryptic growth factors. Control mechanisms, alluded to just briefly in the previous slide. So how do we make sure that we're not just chewing ourselves up into, again, a paste of cells with no matrix? So local activation. Inactive precursors are secreted by individual cells. So plasminogen doesn't do anything, but plasmin actually chews up blood clots. But plasminogen is always flowing around in your body. But it's not until something is generated locally at the site of a wound, in this case TPA, that the clot is actually, the plasminogen is turned into plasmin and can actually do its job. Confinement by cell surface receptors. So cells will oftentimes carry around proteases on their surface, and conformation of the receptor dictates the function of the cell, or the protease that the cell is holding on to. And so what this means is the cells can actually direct these enzymes that are extracellular where they need them. So in this case, for a leading cell, actually moving it to the leading edge where it needs to chew things up. And then secretion of inhibitors. So tissue inhibitors of metalloproteinases, compellingly enough named TIMPs, and serpins, which inhibit serine proteases. So what does this mean for all of us when we're trying to design materials, and how can we take advantage of, it's always a pro and con, especially when you're trying to engineer things. You might say, well, let's just use decellularized ECM because that is all of the structure information, but none of the cells. So if you want to do science on what the structure causes cells to behave, but you don't want like obnoxious immune cells there messing up your experiments, you can get rid of all the cells and just keep the protein structure. And that's actually super useful for some kinds of experiments. But everybody does their decell differently. Some people will use detergents that are really harsh, and you can imagine a lot of the saccharides that swell with water. If you start messing with the ability of water to move around with detergents, it'll really impact the glycosaminoglycans. And it's hard to see reproducibility between different labs doing their decell ECM. Engineering ECM mimetic biomaterials. So there are labs here. If you remember the structure of elastin, that 5 amino acid repeat, there's a lab here, Kyle Lampion, chemical engineering. He actually has bacteria that generate elastin-like protein, and the core of that elastin-like protein that he can synthetically make is the core of elastin. So we can basically make these materials that have the sequences that we know are functional and make them synthetically. So he'll have the elastin sequence, and then he'll probably throw in an RGD in there to make sure that cells can really, really bind to it. So you have high levels of control, but when we look at these large molecules in terms of their most base repeats and kind of ignore the quaternary structure and all the interactions that it can have with all these small molecules, you end up getting something that's non-physiologic. So there's always pros and cons. It's how controlled and how much do you want to know about what your input into the system is. And generally, the closer you are to real physiology, the less we actually know about how it works holistically. I'll close out with a brief example of why integrins are important, and this is just an example of vascular integrity. So this is an outgrowth assay, a bead outgrowth assay. So you've got a gel. In this case, it's a fibrin gel. And you've got a HUVEC-coated bead. So HUVECs are human umbilical vascular endothelial cells. So they really want to grow. but you need integrins in order to have properly functioning vasculature. So if you put in a small molecule inhibitor of, say, an alpha-5 integrin, you actually get no angiogenesis. If you put in a molecule that blocks beta-1, so alpha is one of the two components of a full integrin, put in a beta-1 blocking, you get no angiogenesis. If you put in a beta-3 blocker, you get no angiogenesis. And what it should look like are these guys. So you should get all of these neovessels being formed. But by blocking integrin, you don't get that. And then this is actually intravital imaging. So red here are red blood cells, and green here is an endothelial stain. So the vessels are stained in green, and blood is red. Without beta-1, you actually just get leakier vessels. Vessels don't have the ability to form strong contacts with their basement membrane, which means that they can't really organize themselves into a tube that won't leak everywhere. So dysregulation in integrin function is super deleterious for proper vascularization, especially at the capillary level. I think with that, we're not going to go into the rest of the examples or the super in-depth integrins. So we're good. I will hang around if there's questions. You guys are welcome to come up. But thanks for your attention.